From the phytocypress of the murky black waters of Southeast Asia to the colossal blue whales of the raging oceans, every organism is governed by a predisposed set of genes. Genes carry the biological instruction that defines who we are at the germline level. With over 23,000 genes and about 3 billion letters, the genome unsurprisingly contains some errors that cause biological defects. But what if I tell you that a new technology has been discovered to easily cut, copy, and paste a specific section in an organism's genome just as quickly as in a modern computer? CRISPR is the most talked about and controversial of these biological tools. But what is CRISPR? And how will it propel a new wave in molecular science? CRISPR is a naturally occurring adaptive immunoresponse employed by bacteria. This biological phenomenon was first observed in the 1980s with a bacterium called E. coli. This bacterial immune response is principally comprised of two elements. The first being the alternately positioned repetitive DNA sequences called the CRISPR, which stands for class-third regularly interspaced short palindromic repeats. The second element is the Cas9 protein. This specific protein acts like a biological Swiss Army knife that can cut through targeted viral DNAs. But in 2012, a pair of scientists, Jennifer Doudna and Emmanuel Charpentier, discovered that we could hijack this natural process to modify genomes of any sort. At its basic level, CRISPR-Cas9 will enable us to alter any genes through a laboratory-designed guide RNA bound with the Cas9 protein. The guide RNA will instruct the Cas9 protein to cut the targeted DNA segment, disabling the genome before entering a new one. If pursued further, this gene editing tool will allow humanity to create fungal-resistant crops, edit animals' organs for human transplant, and cure genetically derived diseases like Huntington's and spinocerebellar ataxia. The power of this application lies in its capability to correct any DNA irregularities. However, a powerful thing creates consequential mistakes. The main problem with CRISPR rests on ethical concerns. CRISPR-Cas9 is still a novel technological application that needs thorough examination. But what is clear is that we are already in the dawn of gene editing, where knowledge about the smallest becomes bigger and the impossible quickly unravels.